My name is Zsuzsanna Hollander. I'm Director of Data Science at Genome BC. Good morning, good afternoon, good day, depending on wherever you're calling in from. While we meet today on a virtual platform, I would like to take a moment to recognize and acknowledge that I live, work, and raise my family in Vancouver, British Columbia, which is on the unceded traditional territories of the Coast Salish peoples, including Musqueam, Squamish, and Tsleil-Waututh nations. Welcome to Genome BC's Data Literacy webinar series. We started this last fall, well, actually more than last fall, sorry, in 2022, in fact, with the aim to bring national and international experts together to help deepen our understanding of different data-related initiatives, standards, and resources. Each of our webinars is recorded and available on Genome BC's YouTube channel, so please check it out, the previous episodes. Today's topic, I think, is a very important one, namely is about metadata. And for those of you wondering what metadata is data about data. And you'll hear more about the metadata in different ways that we tackle this. And so in order to address some of the metadata related platforms that are available to you, we brought two presentations, one focusing on protocols IO and another one health metadata commons that was developed by Genome BC. So we'll start first with Dr. Lenny Teitelman, who is the CEO and co-founder of Protocols IO. He has over a decade of computational and experimental biology experience. He did his graduate studies at UC Berkeley, and it was his struggle with correcting a published research method as a postdoc at MIT that led him to co-founding Protocols IO in 2012. Lenny brings to Protocols IO a strong passion for open access, sharing knowledge, and improving research efficiency through technology. Lenny, the floor is yours.
Thank you so much. It's a pleasure to be here. Thank you so much for the invitation and the introduction. I also wanted to give a shout out to Dr. Emma Ganley, who is also one of the panelists and is at Protocols IO. Emma, like me, is a scientist and has been at Protocols IO since 2020, joining right before the pandemic. She's the Director of Strategic Initiatives at Protocols IO, which means that she focuses on working with partnerships with funders and publishers. Before Protocols IO, Emma was the editor-in-chief of PLOS Biology. So I asked Emma to join as well for the Q&A, and because I only have 20 minutes, I won't be able to go into too much detail about if anyone has questions about Protocols IO specifically. So as I present, if you have any questions, please add them to the Q&A. If they're complicated, we'll get to them during the Q&A discussion at the end. And if they're simple or about any aspect of Protocols IO, Emma will be able to reply during the presentation. And with that, I'll share the screen and get started. And in the intro, you already heard about my struggles during my postdoc. And as I turn this on, I'll just say that specifically, it was a protocol on single-cell microscopy in yeast cells. I was doing genetics at MIT, where it took me 1.5 years to change just one small step of the protocol because it wasn't working, and to tweak just a single instruction instead of a microliter of a chemical, changing it to 5 instead of 15-minute incubation in the test tube with it, changing it to an hour. And that 1.5 years to get the method to work, at the end, I realized, oh, it's not a new technique. So I don't get any credit for this 1.5 years. And even more painfully, anybody else who's using this method is going to be getting completely misleading results and has to spend that time, 1 year or 2, rediscovering that tweak. So I sort of became obsessed with creating a central place that makes it easier to share the information about how we do the experiments and how we get the data that we share in our papers. And stealing a slide from Emma, I absolutely love this. This is from chocolate chip cookies, common baking mistakes, a slide from there. It's the same recipe that has produced quite a diverse variation in cookies. Not all of them look great. And the reason we like the slide, the reason I love it is because doing experiments in the laboratory or computational work is very much like cooking. You're going through a set of instructions and looking for a product in the end. And depending on the recipe and depending on how well you follow it and how well the recipe is described, you might get very different results. In the kitchen, it's very different cookies. And in the laboratory, it's very different data. And then you have questions on whether it's reproducible or not. The other, getting a little bit more technical, the other way to look is at an example. This is also not something that I put together. This is from Emerald Cloud Lab. There's a link at the bottom. Hopefully, the slides will be shared after the presentation along with the video. But this is just a tiny section of a methods part of a published paper in a journal from American Chemical Society. And this doesn't look all that bad, but this is not exactly a protocol. This is a little bit more of a summary of what was done. And if you start zooming in, and if you're a researcher who is trying to replicate this work, not just understand what was done in the paper, but trying to repeat it, build on it, take it to a different organism or tissue, you're going to have all these questions pop up, like what exactly does it mean? So you say room temperature, depending on the city, depending on the country and the continent, room temperature can mean different things. In fact, I'll just say that when I was doing my PhD in Berkeley, we were in a new building when we moved, and room temperature instruction, depending on the side of the lab that you were in, if the air conditioner in the summer was blowing in one part of the lab where I was, it was like 4 or 5 degrees colder on my bench than in another part of the lab. So we use these terms frequently in our method section, sometimes even in the protocols, but what's sufficient for understanding what was done in a paper is rarely sufficient for repeating and getting the same results for the reproducibility. And the previous part actually is a good example. That's where you actually have a method section. But for any researcher who has read articles, this is something that's familiar. I actually have bookmarked over the years since we launched in 2014. I've bookmarked statements like this, complaints from scientists, and it would take us probably about 2 hours to go through everything I've bookmarked. So just one example, this is actually from physics. Most of my examples are from the world of biology, but this is a physicist who's reading a 2017 paper, and the method section says devices were fabricated as previously described. Previously described reference from 2015, fabricated as previously described, and the original reference just says devices were fabricated with conventional methods. So you're kind of peeling layers of an onion to try to get at how did you actually do this? How did you generate the data? And at best, the typical paper has a section like this. And at worst and frequently, it is, we used a slightly modified version of a technique reported in another paper, and then good luck. On a more quantitative level, I like the example of a project that happened in 2015 between Center for Open Science and Science Exchange, two organizations that got a grant for, I think, $1.5 million to try to independently replicate in a different lab, independently replicate experimental results from 50 top papers in cancer biology and molecular biology. And there's a total of 190 experiments. And on the summary, so they had a lot of trouble doing this. They were not able to complete it with all 50 papers. And right now, when you go to the Center for Open Science summary of their results, you will notice that 0% of the papers had sufficient detail and the necessary protocol for actually repeating exactly what the other team did. And one of the other things that popped out of that experience is that when Center for Open Science researchers who were trying to repeat the experiments, when they would contact the labs, even when the labs and the professors wanted to collaborate and provide them the protocols, they often didn't know what exactly was done because the student or postdoc who did the work is no longer there. You can't figure out from their lab notebook exactly which version of the protocol relates to the experimental result that's in the paper. So even when you're collaborating with the authors of a paper, it is not trivial inside our teams to figure out what was done 2 years ago in this paper. So we can all do better, both in what we publish and in how we organize and track the detailed research protocols as we do the work, and how we communicate with our collaborators and inside our teams. And that is precisely why we created Protocols.io. The idea we had in 2012, I left my postdoc to do this full time with amazing co-founders. And our mission is simply to make it easy to share method details before, during, and after publication. And what we are aiming to do is, as you can see from the slide, what we're aiming to do is to make it easier to record exactly what you're doing inside the lab, and then facilitate improving the reproducibility and the rigor of the papers that you publish, make it easier to share method details so that we don't have what you saw on the previous slides, have a place, as in my own story, have a place that you can come back to, and update the protocols that you shared in a research paper, so that you can share those tweaks and optimizations, and importantly, have a place where people can get credit for the method development work, because frequently in science, something that should take a week takes you months, in my case, year and a half, and it can be a small tweak, but you should be able to get credit for the work that you've done. So I'll do a 2-minute demo of the actual platform, and then hand it over to Metadata Commons presentation. But at a snapshot, as I said, we launched in 2014. At this point, there are two sides of Protocols.io. There is the public side that I'll focus on for this presentation. That side is open access. It's free to read, free to publish if you're sharing your knowledge openly and publicly. There are no limits, no registration required. And a lot of people, you'll see that there is about 70,000 private protocols. A lot of people also use Protocols.io for that internal sharing and collaboration. That's the part we charge for. So it's similar to the software platform GitHub, where if you're sharing your code repository openly, it's free. If you're using GitHub for internal collaboration, it's paid. For those of you who are researchers funded by Genome BC and have affiliation, you actually have Protocols.io Premium. Genome BC is the first funder in Canada. There's a few in the US. but Genome BC is the first one in Canada to actually provide premium protocols IO, so you can fully use it for internal collaboration and communication around the research experiments and the detailed recipes for them. And you'll be getting an emails very soon with instructions for how to upgrade your account from free to premium. But at this point, in the next 5 minutes, I will do a very quick demo of the key aspects of protocols IO. I'm showing one of those 17,000 public protocols, but I want to emphasize that anything that you create on protocols IO from the beginning, it's secure, it's private, it's yours to start. You decide who to share it with, how to share, what access privileges to give to the people you're sharing with. And if and when you're ready, you can make a protocol public and get credit for it and share your knowledge with others. But it's at your discretion when you publish something and if you publish something. And of course, if you're keeping just internal simple workflows or buffers, simple recipes inside your lab, you don't need to publish everything. It's for sharing the research papers and for getting credit for extensive work. That's when you want to make something public. And the functionality, I want to highlight that these are not just PDFs that are fixed, static text files. The protocols are dynamic and interactive. There's a lot of beautiful functionality that I won't be able to cover on this call, but we have monthly webinars that give an introduction, full introduction to a lot more of the features. We have an advanced webinar happening every month, so you can learn more. But just as a summary, the platform was designed to make it easy to capture the necessary details and aspects of these recipes that will allow your colleagues or yourself 6 months after you've done something to repeat it. And when you make things public for readers of your papers and your protocols to follow up and repeat the work, you can add videos, which really helps for non-English speakers and helps just kind of apprentice style, helps to share how exactly to do something in a way that words do not. And you'll notice on any step of a protocol, as you mouse over it, you'll notice that you can ask a question that will go to the author of the protocol, to everyone who's using it. So for example, on this step number 2, directly on the step, there are discussions between researchers who are using the technique, asking, thank you for sharing this. Can I get a little bit more detail? Where did you order the chemical? Or another question. And so you sort of have back and forth Q&A that's happening, not just by email over and over to different people, but it can happen publicly, or if it's a public protocol, or you can decide to make it private, but you build up FAQs inside your team or on published protocols over time. And as these conversations happen, if you want to clarify anything about your protocol, that part that I mentioned that is important for us, so critical for at the heart of protocols IO, the ability to share an update. You can, with a click of a button, create a new version of a protocol, and the older one doesn't go away, but you can share those corrections, optimizations, clarify things that people are asking questions about. And there is, on every protocol, there is a compare button that allows you at a glance to see which steps are the same, what has changed. So you know exactly what was there before, but you're guided to the most recent version of the protocol. And every single public protocol of those 17,000 that you see on protocols IO is actually a template. So if you're working with brain tissue and I'm working with liver tissue, I could take your protocol. If you've shared it open access publicly, I can take your protocol, make a clone of it. It becomes editable, and depending on the tissues, organisms that I work with, depending on the software language that I use. If it's a computational pipeline, I can tweak your workflow depending on the equipment that I have in our team. And every protocol is at your fingertips, usable in that sense. And for the field methods and wet lab methods specifically, you'll notice that there is a run button, and that's really the last thing that I wanted to highlight that I think is beautiful about, one of the beautiful things about the platform. There is a sort of, going back to that cookie analogy, there is a sort of checklist cooking app that allows you to see, as you're doing the experiment, where you are, what you're doing next. As you go through, you can start a timer on your tablet and a fume hood. And if you change anything as you're performing the experiment, you're not changing the underlying protocol, but you can say, actually, today I did it for 2 minutes or at 6 degrees instead of 4 degrees. And when you save it, you have a record of exactly how you performed the technique on that particular day. So that's another nice feature that helps towards that reproducibility and increased rigor. And I'll finish up by showing one of my favorite examples that you can see here. There's a researcher from Chile who on Twitter is asking for a protocol to get RNA out of cortical neuron cultures. A postdoc from UCSF says, oh, of the 5 that I see on protocols.io, this is the one that should work for you. And what I love about this example is when you look at the protocol, it is in the method section of this paper in the journal GigaScience. And the paper is actually on parasites of stickleback fish. It has nothing to do with cortical neuron cultures, but because the authors shared the rigorous detailed protocol in their method section instead of just that summary that we saw in the beginning, they made their paper more reproducible, and they've put the protocol in a common repository in protocols.io that allows everyone else to build on it and reuse it. And the chance that someone who's looking for a cortical neuron culture protocol is reading a stickleback parasite paper is close to 0. But when we share things in common repositories, whether it's DNA or protein sequences, the genomes, or the protocols themselves, we make them more discoverable and easier to reuse. And the final, next to last slide is, I think I still have a minute from when we started. In terms of getting credit, you can easily share protocols on protocols.io. Love this quote, 80% from a professor in Australia who says 80% of my recent collaborations have been a direct result of sharing our protocols publicly on protocols.io. But when you share your protocols publicly, these are not peer-reviewed papers. We're a platform. It's like sharing your code on GitHub. And it doesn't prevent you from then getting credit and turning your protocols into peer-reviewed published papers. So we actually have partnerships with a number of journals where they welcome submissions from protocols.io to turn your protocols, if you've spent a year getting something to work in crickets that was published in grasshoppers, so that you can get credit for those changes and for that method development work. And the promised last slide is that there are also hundreds of journals, many publishers that explicitly recommend using protocols.io in their author guidelines as you submit a research paper. As you submit a research paper for publication in order to improve the reproducibility and move us away from those vague method sections that leave the readers guessing how exactly would I do it if I needed to repeat this. So with that, I'll share our emails. I know we're not doing Q&A now, but I'll stop my part and looking forward to the Q&A.
Thank you so much, Lenny, for this nice presentation, very informative and with the demo. Our second presenter is from Genome BC. Jojana Hollander is our Director, Data Science. She is an expert with 20 years of experience in clinical research, data mining, and management, database development. She is co-author and co-inventor on many scientific papers and patents on the omics and molecular diagnostic fields. So now, Zsuzsa, the floor is yours.
Thank you, Anaik. So Lenny, I wanted to say that as part of Genome BC's new data sharing and management guideline, we're also requiring protocols.io, so you can add the Genome BC logo onto your last slide. All right, so let's switch gears a little bit and talk about metadata, but in a slightly different context. As you know, there was a huge and rapid advancement in terms of omics research and omics platforms, and that generated a vast amount of data. At the same time, to support the omics research, numerous biobanks have been created housing precious human biological samples. However, today's data silos create barriers to accessing this omics data sets, as well as information about the biobanks that are available in the community. And so to address this need, we have created the Health Metadata Commons, which is a centralized platform that facilitates the access, contribution, and integration of comprehensive genomics research metadata and biobank metadata. We cultivate a synergistic ecosystem where health researchers, clinicians, data scientists, funding entities, policymakers, and other stakeholders in the community seamlessly can discover and request access to pertinent health and omics data alongside human biological samples. Obviously, all of this needs to be done according to what the patient who has provided the samples has consented to. Our overarching goal is to democratize access to genomics data or omics data in general, nurture collaborative endeavors, and expedite breakthroughs within the health sector. By consolidating metadata from Genome BC funded projects and way beyond that, our mission is to empower stakeholders with a unified platform, fostering data exchange, amplifying the visibility of research endeavors, and spurring interdisciplinary cooperation. So we're really hoping to have people use this platform to access the inventory of the omics data sets, and most importantly, to derive additional benefits from the data sets that have already been generated. We think the Metadata Commons has many benefits, and I've only listed a few of them here. We're really hoping that this will lay the foundation for the future of omics research by removing silos through better metadata accessibility and better ways of collaborating. We're also aspiring to have this platform nurture a collaborative knowledge-sharing community, and thus advancing not only the field of omics, but also patient management and with the aging of the population, predictive and other methods to improve human health in general. Through a streamlined data access that's available on the platform, we are aspiring to promote cross-institutional collaboration and thus also improving equity to data for young investigators, researchers from countries with less funding, and others who would like to access data sets that are available in the community already. And with this, I'll go into a demo. I wanted to show here that we have Metadata Commons. You can access it through the .ca, .org, .net, .com website. So you have a variety of ways of being able to access this platform. And so let's take a look quickly as to what exactly it looks like. All right, so as you can see on this site, this is a beta version, and the reason why we decided to release the beta version so that we can gain access, that researchers have access to it and can provide us with feedback early on, so that we can modify it when we fully launch it next month. And so on the first part of the website, on the main page, you see a summary of the different samples. Here, the different omics data sets that are available, as well as the disease conditions that the projects we have currently in the Metadata Commons cover. And as you can see, we have transcriptomics data for almost 2,200 samples. We have 26,000 red blood cells. Whole blood is over 30,000. And so this is already pretty good, considering we're only in beta version and we only have a few projects available at this point. So if all of these are clickable, for example, if somebody's interested in genomics, they can look at the different data sets that are available within that. Or if anybody's interested in doing a different keyword search, that can be done through the Search Metadata Commons tab. And so here you would see the full list of the projects that are currently available, but one could also search for, let's say, proteomics, and then only projects that have proteomics data would show up. All right. So I'll just click on the very first project here to show you the type of information that we're asking for. Some basic information about the projects, who funded it, who are the investigators, and if there is a key publication that can provide additional details, that could be also posted here. Some aggregate level information about the patient cohort, where the enrollment was, how many subjects were enrolled, what the disease studied was. And then in terms of clinical data, there is no patient level information whatsoever. We just ask for the type of clinical data that was collected. That way we are keeping patient privacy and confidentiality. Then next we have information about the samples and the associated omics. As you can see here, for example, the serum wasn't analyzed, but we would still like to have that information in case somebody is interested in those serum samples and would like to ask the researcher for a collaboration to gain access to those samples and profile them using, let's say, metabolomics. We also have the contact investigator button here that will pop up on email where you can customize, put in the information, what kind of project you have in mind, what kind of data you would be requesting, do you have ethics approval for this study, and so forth. And that email will go to the investigator, and they can get back to you about information, whether they can collaborate, and if the patient consent covers the type of questions that you would like to ask of the data. There is also a metadata submission tab. And so for this, one has to log in order to access the metadata submission form. And here I would see all the different projects that have been submitted by me, but I can also start a new submission and enter the information that we have already gone through. One of the features that we have also added for users who choose to create an account, they can log into the account and set their preferences. So what does that really mean? If you add information about a study that you're interested in, let's say I'm interested in only clinical trials that have collected, I don't know, maybe cord blood, and interested in a specific omics type, you can apply these changes. And then when a data set satisfying those criteria becomes available, you would be notified by email that's on your user account of the different data sets that meet those conditions. So you don't have to wonder, oh, I wonder if there is any new data sets available that I'm interested in. And you can update these preferences as often as you need as your research interest changes. We also provide additional resources here. And one thing we have added was biodiversity data sources, as we would like to expand beyond the health sector. We would be adding additional information for sectors outside of health, since Genome BC is also supporting the agri-food and natural resources sector. And with this, I'll stop sharing my screen, and I'll be happy to take any questions.
Oh, thank you very much, Yusha, for this great presentation. We have a few questions, and the first one is about both protocols IO and metadata commons. And the question is, how secure is the protocols and metadata servers? Lots of data breaches these days. Yusha, do you want to start?
Sure, I could start. Yes, so in terms of data bridges, as you could tell, there is really no patient identifying information that we are storing on the platform. It is on the Oracle Cloud solution. And so all the information is publicly available. There is nothing in there that would be worrisome. The only thing is the user accounts, but through the Oracle solution, we have security ensured. So data breaches are hopefully preventable through that. Although these days with the cybercrime advancing, you can never say that everything is secure, but we do everything that's possible to keep the user accounts information safe.
Thank you, Yusha. Lenny?
Yes, and from the protocols IO side, we are just absolutely paranoid about this. We have pharmaceuticals that are using protocols IO, so we've been, you can only do so much, but so far, knock on wood, we have not had breaches. We're using Amazon Web Services and their security. We have two-factor authentication. We have invested a lot into penetration tests and policies internally that allow research hospitals to use us, Food and Drug Administration in the United States, pharmaceuticals. So it's not just the publishing. We know that you're storing information that you might not want to make public. We don't take that for granted. So we have put a lot of effort into the security, and on Protocols IO, if you go to the help and FAQ, you can see our security measures. There is a security white paper that describes our procedures and approaches around this. And Emma, maybe we can put a link in the answer directly to that, but great question.
Thank you, Lenny. A question about metadata commons for Yusha. Is there a curation process for reviewing and updating metadata entries over time?
Yes, so one thing I didn't show due to time limitation was that all the metadata that's submitted comes to us, and the administrators in the background review it to make sure as much as possible that the data is accurate and complete. And so only once the project has been approved, it is published on the Metadata Commons website. In terms of updating metadata entries over time, if the user requests an update, we can reopen the project, and through their own account, they can update any of the entries that have changed. So for example, if all of a sudden, for the example that I've shown, the serum samples happen to have been profiled since, they can add the omics profiling that was done on those samples, and then they can resubmit it and goes live again with the new changes.
Thank you very much, Yusha. There is another question about, do you store the data on the cloud? And if so, what about jurisdictional restrictions?
So I'll start on our side, and then Lenny can answer from the protocols IO side. We only store metadata. All of it is on a cloud solution, the Oracle Cloud Solution, as I mentioned before. And because of that, there is no jurisdictional restrictions, although the metadata itself is on a Canadian cloud solution. Lenny, for protocols IO?
Yes, that does come up. There are instances where we need to put a server in the location of the research organization that is using protocols IO, but Amazon makes it easy to do that. We're GDPR compliant, and actually, since we're acquired almost a year ago, plus minus 10 days by Springer Nature, they're legal, did another pass through our terms of service, through GDPR compliance, and we've been making sure that we're compliant with the requirements and doing everything we can to protect the data. And you know, that information, I think, is all available on protocols IO, but it's a good question. And the jurisdiction parts, when we do need to host the data somewhere else, the user information, the accounts and passwords, that does add complexity, but we are set up to do that.
Thank you, Joja and Lenny. The next question is, are there standards or guidelines for metadata formatting and annotation within the commons?
Yes, so this is the first version. So, you know, clearly it's not going to be perfect from the very beginning. We're in the beta stage, and as I said, we really welcome input on this. However, we have started with, for some of the variables with ontologies, for example, the disease condition. We have a disease ontology in the background. And so when somebody starts typing in a specific disease category as they're entering the metadata, the options pop up, and then you can choose from those. We have created standardized data entries for the omics subtype, and a few of the variables that were sort of things that we wanted to make sure that are not going to be kind of chaotic in the background. Some of these variables, the users can also add a new, let's say there is a new platform that we haven't encountered yet. A user can add it, and in the process of approval, we can also review those and make sure that there is no duplicates.
Thank you, Joja. The next question is, are there opportunities for collaboration or partnership with external organizations or researchers to enhance the metadata commons?
Yes, thank you. I love this question. Over the past few months, we have been presenting at various research institutes. I'm going to the Festival of Genomics and Biodata, actually leaving tomorrow. And so one of the things that we've been focusing on is please come and work with us and help us enhance the metadata commons. We really welcome collaborations. We welcome others to come in. I would especially love to work with trainees who are eager to contribute to the field of the metadata. And yes, please contact us at metadatacommons@genomebc.ca, or you can find my email address on the Genome BC website. Please reach out, and I would love to work with you.
The next question for Lenny. Are most the protocols US-based, or do European researchers and from elsewhere also use protocols IO, and do they have something similar?
So I will actually ask Dr. Ganley to answer that, because She is in Europe. I am in US, but she is in Europe, and she's best positioned to address this.
We have a lot of content actually globally beyond Europe and America or Canada. And I think it's probably about 1/3 rest of the world, 2/3 US and Canada right now. But that is expanding all the time. And the majority probably of other content is within Europe. But there is significant content across Australasia and out with the main continents, I think, too. And we're working with some institutions in Asia as well, which is really nice to see the uptake in other places where they don't have always access to our content. So yeah, very much global, with right now probably more content North America, Canada.
And Emma, can you also, part of the question was, is there something similar outside of US? Do you want to say a few words about Protocol Exchange, maybe, because that is another repository?
Okay, sorry, I missed that nuance. There is another repository that was also started by Springer Nature, which actually predated protocols.io, called Protocol Exchange. The functionality never really grew there, so very much the content was quite static as PDF format, more as a journal article would look. And since our acquisition by Springer Nature, they have made the decision to sunset and close Protocol Exchange to new submissions. And so that has now happened, and we are gradually moving all of the content that's been published on Protocol Exchange over onto protocols.io, so it will become more dynamic. It was already possible to version on Protocol Exchange, but that is as far really as the functionality went in terms of it being dynamic. So now the content will all have more dynamic functionality with all of the whistles and bells that Lenny showed you. And that was probably the biggest kind of equivalent platform previously, which didn't fully have all of the same functionality. So we still believe we're the main option for adding protocol content, posting, publishing, and having all of this dynamic functionality.
Thank you very much, Emma, and thank you for answering all the questions live as well. The next question is for Lenny. Are you going to implement AI to help find protocols?
We actually have a public roadmap that we share, development roadmap, and that is one of the 5 things we're focused on this year in terms of discoverability of protocols. And that is an area where we think AI can shine in terms of finding related methods and giving you the technique. That's always been a challenge. In the early days when we had 10 total protocols in 2014 that were public on protocols.io, there was no need to search. You just saw all of them on one page. Now that there's 17,000, and we're getting more from Springer Nature, like Emma described, in terms of the Protocol Exchange, and there's another related question about it, it's only going to become more and more of a challenge. And so that is a big priority. I don't want to overpromise, but that is on the roadmap and is an important focus this year.
Thank you, Lenny. The next question is for Lenny and Emma. Does protocols.io also invite protocols in the realms of social sciences and other protocols other than in the lab?
We absolutely do accommodate protocols in the social sciences. It's less commonly used that way, but we do have some of the institutions that we work with have very specifically asked us to give webinars on how you could use protocols.io for those disciplines. So we see across all areas, actually, so psychology, sort of ecology applications, which you can put sort of questions and questionnaires in a limited way, but because it's such a bare template structure, if it has some kind of process format that is a step-by-step approach as a part of any kind of topic, you can put it in. And there is a similar concern around reproducibility in the social sciences. It's framed slightly differently, but it's there and it exists, and it's something that they're conscious of and do want to try to address also. So I'm really interested to see how that grows. I think we're just seeing more use in those areas.
Thank you very much, Emma. That's great. One question that has been answered live, but we'd like to highlight again. So how does the Protocols.io platform allow researchers to get publication credit? Lenny?
Will I take that? Oh, you want me to take that? Sorry. You can fill in if I miss anything. So because you can post your protocols publicly and get a DOI, it's very much like a preprint when you post that way, in that we do a cursory screen, but we don't peer review. But once the content is published and made available, that kind of ongoing review happens by other scientists. And that is then an independent output. So the protocol itself is citable. If you link your account with your ORCID profile, for example, it will appear as a cited, as a kind of a work on your ORCID profile. They're also indexed by places like Google Scholar, and we're working with Europe PubMed Central. So, and Lenny's just nudging me towards talking about peer review options. When you have that DOI, you can put that into a research paper that you publish. So it's very, very easy to reserve a DOI or publish it. If you don't want to make it publicly available until a corresponding paper is published, that's what that reserved DOI option is for. And then there are peer review options. We have very close relationships with a handful of journals right now, where PLOS One Lab Protocol actually requires inclusion of methods on protocols.io. The other two journals just strongly encourage using protocols.io for any relevant protocol content. And in those journals, the protocols on protocols.io will be peer reviewed with the paper. And we can actually add a badge to reflect that they're peer reviewed in those situations. Beyond those three journals, well over 500, probably more like 1,000 post Springer Nature acquisition are very happy for you to put the methods link to a protocols.io in the materials and methods of the papers that you're publishing. So it's growing understanding that the method output is an important standalone research output that deserves recognition. You can have a different author list. So maybe the people who develop the method are different than the people who apply the method to a research question. So you can actually reflect that with a different author list as well on the methods on protocols.io.
Thank you so much, Emma. There is one question about the metadata commons. The question is, the metadata commons is still in beta version. What is the vision of what that roadmap would look like?
Thank you. So we, as I mentioned, we posted the beta version for anybody to test it so that we can have feedback. We are going live next month. And currently, we're trying to find additional ways to attract data. We're reaching out to researchers that we have funded or who we know in the community, doing a lot of outreach. But we also have plans to go out to the public repositories and contact investigators who have already provided their data in those repositories, so that we can list them in the metadata commons. One of the benefits, I think, of the metadata common is that we list not just one specific omics type, like the repositories that are usually concentrated around one specific omics. And so researchers these days are quite interested in doing multi-omics research. And by using our platform, one can find projects that have more than one omics data type already available, thus supporting those kind of collaborations where multi-omics research can be done. And so we have pretty ambitious plans to attract metadata from sort of worldwide publications and researchers. And if anybody's interested in bringing in their projects, we really would welcome you. If you have ideas as to how to promote and attract additional projects to be listed, please let us know. We really, truly welcome any feedback. And so please contact us.
Thank you so much, Zsuzsa. And that was our last question. Thank you again to our speakers, Emma and Lenny and Zsuzsa. Thank you everyone for your attention and for joining Genome BC Data Literacy webinar today on the power of metadata. Please visit the events.
Sorry, I just wanted to give a shout out to Anaik, who worked alongside me for the past almost year and a half on creating the Metadata Commons, along with many of my colleagues, including Federica, our CSO and VP of Research and Innovation, who has been instrumental in getting this up and running, or an executive who allowed us to do this and develop it, and hopefully support many collaborations over the next few decades or years in the omics space. And thank you, Lenny and Emma. This was great to hear about Protocols.io. And in terms of the Protocols.io, if you have been funded by us, or if you have an account and you work with Genome BC, you will get an email soon that will allow you to upgrade your account. You can also either email Protocols.io or us to ask for an upgrade code in the meantime, if you're too eager to get going. So please use these tools because it's going to benefit your research and it's going to benefit others as well. Thank you so much.